Hello. I'm Shira.
I'm Sigan.
I'm Grace. I'm Lisa.
We are from Group C.
History.
We are going to present
Signal transduction mechanism 1.
In signal transduction mechanism, we are going to focus on mediators and second messenger system. In mediators, we are going to learn about first messengers, second messengers, protein kinases, G proteins, cyclic guanosine monophosphate, cyclic adenosine monophosphate. Whereas in second messenger systems, we are going to see about adenylene cyclase and cyclic adenosine monophosphate, calcium ion, phospholipase C, diacylglycerol, and inositol triphosphate.
So now, I'm going to explain the mediators that involve in the signal transduction pathways. The first messenger is this extracellular molecule that binds to the specific molecules on the target cells. For example, peptide hormones, neurotransmitter, and the epinephrine. The first messenger also can induce the production of second messenger. Next, the second messenger is the intracellular molecules that enter or generated in the cytoplasm. For example, is the cyclic adenosine monophosphate. While the protein kinases is enzymes that transfer a phosphate group from ADP to a protein through phosphorylation.
Next, we are going to see G protein and cyclic adenosine monophosphate CAMP and cyclic guanosine monophosphate CGMP, which are categorized under second messenger. G protein is three-part membrane transducer molecule linked to cytoplasmic tail of a protein, which is bound to guanosine diphosphate GDP, which is inactive, and guanosine triphosphate GTP, which is active. CAMP is a product of cytosolic ATP molecule, which is catalyzed by membrane enzyme called adenylyl cyclase. CAMP activates CAMP dependent protein kinase, which is known as protein kinase A. Basically, CAMP is a product of cytosolic ATP, which is catalyzed by adenylyl cyclase, and it activates CAMP dependent protein kinase. Next is CGMP. CGMP is product of cytosolic GTP, which is catalyzed by membrane enzyme called guanylyl cyclase. CGMP activates CGMP dependent protein kinase, which is known as protein kinase B. To sum up, CGMP is a product of cytosolic GTP, which is catalyzed by guanylyl cyclase, which activates CGMP dependent protein kinase or protein kinase B.
One of the second messenger system is cyclic AMP pathway, and we're about to discuss the mechanism and also importance of it. So here is the mechanism of the cyclic AMP pathway, and we have choose adrenaline to be the first messenger, stimulatory G protein to be the signal transducer, the adenylyl cyclase to be the primary effector, the cyclic AMP to be the second messenger, and the secondary effector is protein kinase A. So firstly, when adrenaline binds to membrane receptor, stimulatory G protein is activated, and then the alpha subunit of stimulatory G protein will stimulate the adenylyl cyclase to convert ATP into cyclic AMP. And after that, the protein kinase A is activated and phosphorylate many different target proteins, which results in cell's response. Okay, so next, there are two importance of this kind of second messenger system. The first one is original signal amplified in each step to produce large effect. For example here, from one molecule of adrenaline will become 100 molecules of activated G protein, and later on become 1000 molecules of cyclic AMP, and so on. Okay, other than that, cyclic AMP dependent protein kinase can phosphorylate many different proteins. For example here, it may go to target proteins in cytoplasm or diffuse into the nucleus. So that's all for this part. Other than adenylyl cyclase and cyclic adenosine monophosphate, calcium ion also serves as the second messenger. Calcium ion can enter the cytosol from two sources, which are the extracellular fluid and the endoplasmic reticulum. From the extracellular fluid, the calcium ion can enter the cytosol through voltage-gated calcium ion channels or the ligand-gated calcium ion channels. From the endoplasmic reticulum, the process is induced by a second messenger, which is known as the inositol triphosphate. It can activate the opening of the calcium ion channel. Electrochemical gradient favors the movement of calcium ion into the cytosol. The increase in the cytosolic calcium ion concentration can be achieved by inhibition of active transport of calcium ion from the cytosol into the extracellular fluid and increase the membrane permeability to calcium ion. Calcium ion involves two mechanisms, which are the stimulation of a cell leads to an increase in cytosolic calcium ion concentration, and also an increase in the cytosolic calcium ion concentration induces a cell's responses. Let us start from the first mechanism. The first mechanism is the stimulation of a cell leads to an increase in the cytosolic calcium ion concentration. In response to a signal molecules, the cell surface receptor protein activates a G protein, which in turn activates the enzyme phospholipase C. The enzyme catalyzes the production of inositol triphosphate, which diffuses through the cytoplasm to the endoplasmic reticulum. Inositol triphosphate binds to and opens the calcium channels, allowing the calcium ion to flow from the endoplasmic reticulum into the cytoplasm. Calcium ion triggers the protein sensitive to calcium ion to initiate a variety of activities. The active transport of the calcium ion out of the cell is inhibited by a second messenger. Now, let us move on to the next mechanism, which is the increase in the cytosolic calcium ion concentration induces the cell's response. Calcium ions bind to the calmodulin. On binding calcium, the calmodulin changes its conformation, which allows it to activate or inhibit a large variety of enzymes and other proteins. Many of these enzymes are protein kinases. Calcium ions also combine with calcium-binding intermediate proteins other than calmodulin. These proteins then act in a manner analogous to calmodulin. Sometimes, calcium combines with and alter the response proteins directly without the intermediation of any specific calcium-binding protein. So now we are going to discuss about the second messenger system that involves the phospholipase C, the diacylglycerol, and inositol triphosphate. So the phospholipase C is a membrane-bound enzyme, and the PIP2, which is phosphatidylinositol bisphosphate, is a phospholipid component of the cell membrane. When the signal protein binds to the G protein link receptor, it will activate the G protein, which is following by the activation of the phospholipase C enzyme. The phospholipase C enzyme will then hydrolyze the PIP2 into the diacylglycerol DAG and the inositol triphosphate IP3. The diacylglycerol will remain on the cell membrane and then activate the protein kinase C. On the other hand, the inositol triphosphate will then diffuse into the cytosol and activate the IP3 receptor on the endoplasmic reticulum membrane, which is a particular calcium channel. This will cause the calcium channel on the endoplasmic reticulum to open, and then the calcium ion will diffuse up into the cytosol. The calcium ion will eventually activate the protein kinase C. The protein kinase C will in turn activate other cytosolic protein. This will cause a cascade of signal pathway, which will result in the cell's response.